Okay, guys, so we are going to start here. This is pretty much where we finished off on Friday. And so basically, we're just a quick summary of what the action potential looks like. Remember, we start with resting potential. This is when we have active transport of sodium and potassium pumps, keeping the concentration gradients with sodium high on the outside and potassium high on the inside. And then we have a neuron stimulated, and this causes a depolarization. This is when the voltage-gated sodium channels open up and allows sodium to flood into the axon, and this flips it from being negative on the inside to positive on the inside, which you can see here. At resting potential, we have positive on the outside and negative on the inside. Depolarization flip-flops these two because sodium, which is positive, comes rushing in, so now it's more negative on the outside. This graph, if you're looking at it, is really showcasing the charge within a membrane, within the axon, okay? It's kind of happening down here. This is the axon here, okay? Now, step 2 is depolarization, so we jump up to a positive charge inside the axon. Step 3 is repolarization. This is when the voltage-gated potassium channels open up, allow potassium to flood out, and therefore we have a reversal of charges back to where it was. So we now have positive on the outside and negative on the inside once again, but the sodium and potassium are on the wrong sides. We're supposed to have potassium on the inside and sodium on the outside, but this has been flip-flopped. So we have to go back into resting potential. Oops, sorry. We have to go back into resting potential in order to get the sodium and potassium back on the right sides. Let's take a look at this through the McGraw-Hill animation.
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in, while other channels allow some flow of potassium ions back out of the cell. The sodium ions cannot easily get back in to replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside the cell.
Okay, so what you can see here, this is what the impulse looks like. It's going to travel down the axon, and behind it, it's going to start going back to resting potential so it can reset. So it kind of sends it in a wave where we have enough stimulated neuron here to allow it. This allows the sodium channels next to it to be stimulated and allows the sodium channels to open just behind it. So that's depolarization in purple. Repolarization is happening here in green, and that's where potassium is allowed out of the cell. And then just after that, we have resting potential again. So it sends it through the membrane like a wave. Check it out.
Membrane potential, and to reestablish proper concentrations of sodium and potassium ions inside and outside of the cell.
Okay. So that's what's basically happening here. Let me talk to you about what these phrases mean. The threshold here, this is essentially when, we're going to talk about it more, but when the threshold is reached, this stimulates the sodium channels to open. When enough sodium channels have been opened, this is going to cause the ones next to it to continue opening. This is known as action potential, okay? This is sodium running in and depolarizing the region. Just after that, we have repolarization, okay? And we have the refractory period. So from here on, we have repolarization and then resting potential after that. The movement of which ion initiates an action potential? Is it calcium, magnesium, sodium, or potassium? The correct answer was sodium. The movement of sodium initiates the action potential. Having those sodium gated channels open and have sodium flooding in, this starts the whole process of action potential. Okay, this is what we're going to look at one more time, just in notes form rather than me just explaining it on a video. But depolarization happens in response to a signal. The axon becomes depolarized. This is the second step. We talk about the first step being just resting potential. But that particular area gets depolarized. One or two sodium channels open. This causes the whole region in that area to open and become depolarized. You'll notice the rest of it has not been depolarized. This is going to shift down and continue moving towards the end of the axon terminal end here. And basically, the next step is repolarization. So this is when the potassium comes flooding out of the axon, allowing the charges to flip back to the way they belong. Depolarization is going to continue moving this way. And repolarization, the charges are going to get flipped just behind it. And then finally, we're going to go back to resting potential just after that. As this gets depolarized, this gets repolarized, and then we go back to resting potential. Please understand that this is one of your understandings. So basically, the propagation of nerve impulses is the result of local currents that use each successive part of the axon to reach the threshold potential. Look at the action potential propagation in an unmyelinated axon. We talked about myelination at the beginning of the notes. That's myelination. When a nerve is myelinated, it's wrapped in the Schwann cells. When it's unmyelinated, that just means that it's nice and smooth and continuous action potential. Check it out.
An action potential, depicted as a red band, is propagated in one direction along the axon. During an action potential, the inside of the cell membrane becomes positive with respect to the outside. An action potential generates local currents that tend to depolarize the membrane immediately adjacent to the action potential. When depolarization caused by the local currents reaches threshold, a new action potential is produced adjacent to the original one. Action potential propagation occurs in one direction because the recently depolarized area of the membrane is in absolute refractory period and cannot generate an action potential.
Okay, that refractory period is when it's essentially resetting. So that's just showing you the direction that it travels in. For animation, which is right here, if you click on it on the notes I provided for you on Canvas, if you're still struggling to fully understand, this is kind of a comprehensive video to showcase everything that we've talked about. If you are struggling, I highly recommend you go to the notes, click on this link, and watch that animation. Okay, now going back to myelination and saltatory conduction, we talked briefly at the beginning about the Schwann cells, which are featured here and here. Okay, you can really see that wrapping here. A Schwann cell is a specialized cell that basically acts as a insulator. Okay, this is it is also called a myelin sheath. So the Schwann cell, all the Schwann cells build up together, create what's known as a myelin sheath around the axon. And this allows the signal to go really, really fast. So think about the nerves that you would want to have a very fast impulse traveling through, up to 200 meters per second along the myelinated axon compared to unmyelinated, which is just 2. So 200 to 2, it's much, much, much faster. And that's crucial for reaction time, all sorts of different things, okay? Now what happens is the action potential jumps from each node to each node of Ringvier. It's going to jump, and this is what speeds up that process, okay? This is known as saltatory conduction. It reduces the degradation of the impulse as well, so that it travels really fast and really strong. It also reduces the amount of energy needed to have that signal travel across. Think about all of the active pumping that we have to do to maintain this concentration gradient. We don't need to do that in the areas underneath the cells. So it does a couple of things. Let me erase this. It will make it travel much faster, okay, which is here. It reduces the energy necessary to have a signal passing, and it allows it to degrade, it allows it to be a lot stronger. So 3 reasons why myelination is pretty important for our nerve cells. Okay. On your Edpuzzle, please explain how a nerve impulse passes along a non-myelinated neuron. Now, I'm looking for neurons that are not insulated with Schwann cells. How does this work? So this is everything we've been talking about except for this last slide, which was essentially all about the Schwann cells, the nodes of Ringvier, and the saltatory conduction. So talk to me about that. 8 marks. So here is what the moderator was given. Take a minute, look at it, and see how you did in comparison to what the moderator was looking for.